Why, hello there! Welcome to the exhibit on the identification and characterization of head inducers. This exhibit was created by Chen, as pictured here behind me. I am Mr. Stickman, but you can call me your man. And today's tour, please follow me as I guide you through our fine exhibit. Our story begins with these two famous German embryologists, Hans Spemann and Hilda Mangold. In experiments that were published in 1924, they found an area in an embryo that, when transplanted, induced or organized the development of an entirely new axis, resulting in effectively twin conjoined larvae. This region of the dorsal lip would become known as the Spemann organizer. The experiment involved the surgical excision of the Spemann organizer, or the dorsal lip, from the donor embryo, to be transplanted into the ventral end of the host embryo. The transplanted cells from the donor made up only the new body's notochord, while all the other new developments were induced by the organizer from the host's original cells. The organizer organized the surrounding cells and directed them into the development of an entirely new body axis. Later experiments would go on to show that different parts of the organizer would induce different parts of the body. This landmark discovery started the quest to identify the inducers, the chief quest being the discovery of what was, or were, the neuronal and head inducers. The quest to find these inducers took many, many years. It wasn't until 1995 that it was discovered that BMP4 induced epidermal fate, and that neurons were the default state. It was soon then found that neuronal inducers, really BMP4 inhibitors, were chordin, noggin, and follistatin. With neuronal inducers beginning to be characterized, head inducers would come very soon afterwards. T. was Bowister and V. Robertus at the Howard Hughes Medical Institute of UCLA in 1996 published a paper on their discovery of Kerberos. It is a secreted factor of the anterior endoderm of Smemann's organizer that they believed induced head development. In their experiment, they found that when Kerberos mRNA was injected into the V4 ventral vegetal blastomere, an early embryo, it formed an ectopic head. Kerberos was found to be sufficient to induce these secondary ectopic heads, complete with head structures like brain gray and white matter, olfactory placodes, and the eye. It was also sufficient enough to induce other anterior mesoderm structures, such as the cement gland, heart, and liver. These secondary heads also lacked any trunk-tail axial structures, like somites or notochord, as well as an eye. The ectopic heads were cyclopic, having only one eye. This was proposed because Kerberos inhibits prechordal plate function. It was further found that Kerberos can induce neuralization and was antagonized with BMP4. These results led Bowister and de Robertis to conclude that Kerberos promoted the anteriorization of the embryo while inhibiting trunk-tail mesoderm formation. 2 years later, Glinka and Neers in Germany found yet another head inducer. They found a new family of secreted protein, DKK. DKK1 functions as a head inducer and is also expressed by the Spemann organizer. They found that DKK1 with BMP4 inhibition via co-injection with dominant negative BMP4 receptors, TBR, mRNA would result in complete secondary axes with two eyes, cement glands, and two beating hearts. This result led to the conclusion that DKK1 anteriorizes embryos and is able to induce head structures when working in synergy with these BMP4 signaling inhibitors. DKK was further characterized by its ability to inhibit Wnt signaling. DKK1 was able to rescue a xenopus that had a secondary axis induction caused by an overexpression of Wnt. Co-injection of DKK1 with Wnt resulted in a normal xenopus. DKK was also found to be able to inhibit late-action Wnt8-2. In both cases, DKK operated extracellularly. Glinka and Neers also show that DKK is necessary for head induction via antibody inhibition experiment. Injection of anti-15 antibody led to severe microcephaly, which then could be rescued by DKK1 injection. 1 year later, Stefano Piccolo, who worked in De Robertis' lab at UCLA, further characterized Kerberos while also putting Kerberos and DKK1 together to further elucidate the process of head induction. He found that the Kerberos protein is a trifunctional antagonist that binds to XNR1, otherwise known as nodal, BNP, and XWNT8, otherwise known as Wnt, in solution. All three signals, nodal, BNP, and Wnt, must be inhibited in order for head formation. Further, they found that endogenous Kerberos transcription is turned on by early nodal signaling. Thus, we arrived at a process. Head induction is accounted for by cordon inhibiting BNP. DKK1 will inhibit Wnt. Early nodal signaling is required to turn on transcription of Kerberos, which when made will bind nodal, BNP, and Wnt, locking the head organizing program in place and restricting the trunk territory to the posterior part of the body. And thus, we have head induction.